Hello, good afternoon, everyone, and welcome to the BES-Net TT project webinar. A special welcome to everyone who's joining us for the first time on this platform. My name is Celeste Shariandi, Science Communication Officer for BES-Net TT. BES-Net TT is the acronym for the Biodiversity and Ecosystem Services Network Trinidad and Tobago project. This is a project which is being implemented by the United Nations Development Program and the Ministry of Planning and Development through the Environmental Policy and Planning Division. The project has a focus on pollination and pollinator management and has three components: science, policy, and practice. Under the science component, the project undertook a seagrass survey to investigate seagrass flowering and pollinators at two coastal areas, one in Trinidad and the other in Tobago. The research activity was conducted over the course of a year by a small team led by the principal researcher, Dr. Kelly Kingon, who's right there and with her camera on. And the findings of this research activity are being shared publicly to build awareness of the pollination of seagrass species and fill research gaps for this ecosystem. It is hoped that the new knowledge can contribute to key recommendations for pollinator management. Before I hand over to Dr. Kingon, here are a few housekeeping rules for the webinar. This session will be recorded on Zoom, and the recording will be posted on the BES-Net TT Facebook page, probably later today, and uploaded to the BES-Net TT YouTube channel. In order to keep within our 1 hour time frame, the presentation will take approximately 30 minutes and will be followed by a 15 to 20 minute question and answer segment. Essentially, however much time we have after the presentation, we can take questions. While the presentation is being delivered, you may post any questions that may arise in the Q&A area of the Zoom platform. If the question and answer segment, if during this segment it is not possible to answer some of your questions, these will be addressed afterwards, and the answers will be posted with the recording of the session on the BES-Net TT project Facebook page. So now I'll introduce our presenter. Dr. Kelly Kingon works at the University of Trinidad and Tobago within the Marine Sciences Unit as an assistant professor and discipline lead for the MSc program in integrated coastal and ocean management. Her research focuses broadly on marine conservation issues, including threatened species, declining habitats, introduced species, and the impacts of parasite prevalence on important reef herbivores. Dr. Kingon has studied seagrasses for over 15 years and is excited to share her knowledge on them and what was found in the BES-Net TT research study on seagrass flowering and pollinators in Trinidad and Tobago. I now hand you over to Dr. Kingon.
Thanks, Celeste. Good afternoon, everyone. Thank you so much for coming today. My name is Dr. Kelly Kingon, and I'm an assistant professor at the University of Trinidad and Tobago, as well as a director of the nonprofit organization SHOPO. First of all, I just want to draw attention to all the assistants that I had with this project. People are all listed here that helped in the field or with the lab work or data entry. I had a lot of assistants, and I couldn't have done it without all of these fabulous people. Next, I want to thank BES-Net for involving me in this project. It's been a whole lot of fun. And of course, the funders, who consist of the governments of Trinidad and Tobago, UNDP, the government of Germany, through the International Climate Initiative and SwedBio. And also want to thank the Tobago House of Assembly and the CDA for granting us permits to do this work in the locations where we're at. And finally, I want to thank Bridget Van Tossenbroek and her colleagues, including Ricardo Wong, who kind of helped with the initiation of this project on seagrass pollinators. So to start, I want to provide a little background information on seagrasses, their importance, and how they reproduce. So seagrasses are submerged marine flowering plants. They live in clear, shallow water in coastal areas that are protected from wave action, either via being an embayment or protected by an outer coral reef. They are found in tropical and temperate regions across the globe. So why should we care about seagrasses? They're important nurseries for many fishery species, including shrimp, snappers, grunts, groupers, jacks, lobsters, conch, and more. Their roots and rhizomes, which are their horizontal stems, help to stabilize sediments, prevent erosion. They protect the shoreline, and of course they produce and hold a lot of organic matter that provides a huge carbon sink. In addition, the blades help to slow currents. The leaves also filter water of nutrients and toxins and other things. And with the roots, they support a wide diversity of life. Seagrasses are also direct food sources for many animals, including green sea turtles, herbivorous reef fishes, sea urchins, snails, and manatees. And they're an indirect food source via their detritus, which is the dead decaying plant matter as the leaves die, and the epiphytes, which are tiny little creatures that grow on the blades of grass. Many things eat those components of the seagrass if they don't eat the seagrass themselves. Seagrasses have very high rates of primary productivity. They're important for nutrient cycling. And of course, they produce a lot of oxygen and they help to decrease the carbon dioxide. So how do seagrasses reproduce? Most seagrasses can reproduce both sexually and asexually. Asexual reproduction happens via rhizomatic or horizontal stem, those horizontal stems again, which you can see in the diagram on the right. So those rhizomatic stems grow and expand the grass throughout a whole bed. And that makes one clone, right? It's all the same individual, throwing up additional ramets, which are kind of each little section of leaves that comes to the surface. Those would be called ramets along the horizontal rhizome, which is here. And then the roots coming out here, right? They also can asexually reproduce via fragmentation. So these fragments can break off with storms or with grazing, like you see with these green sea turtles grazing here. And via human impacts like anchors or prop scars, things like that. And so when a little piece of this rhizome with leaves attached or not breaks off, and then it can float away and then sink in a new location and begin to grow in that place. So that's one way that they can kind of increase genetic diversity asexually. So these seagrasses can form really large clonal meadows. So recently they found a clone of Posidonia oceanica, Neptune grass in the Mediterranean Sea that covers 15 kilometers, and it's all one individual. And it's likely that this clone, this individual is thousands of years old, which is pretty cool. So if the environment is relatively stable, these clonal forms like this can work really well. Fragments from this large clone can spread and move into other areas, which would increase the genetic diversity in the other areas. These clones are heavily adapted to the local conditions, but with all the human disturbances going on, it can upset this balance. And when that happens, you really want higher genetic diversity, not just one single clone, so that some members of the population may be better suited to cope with these increased disturbances. So in order to create more genetic diversity, seagrasses can reproduce sexually as well, and they do this via flowers. The flowers are typically dioecious, which means that male and female flowers are on separate plants, but there are some species, and some that I'm not going to mention today, that are monoecious, which means male and female flowers on the same plant, and others that are even hermaphroditic, which basically means the same flower has both male and female parts. Okay, so typically it was thought that pollen just dispersed via water movement for these different seagrass species. So the male flower would release the pollen, it would float by currents, and hopefully end up on a female flower somewhere downstream. The pollen, to best suit this type of pollination, is typically elliptical and long in shape, but some of it can be more spherical, like pollen that you would find on land that typically travels by wind. Often the pollen is contained in a sticky mucilage, or kind of like sticky stringy bits. And this helps to facilitate once the currents hopefully pull the pollen to the female, but then it will stick to the female once it sinks down. But we're also finding that this sticky mucilage and these sticky strings are also being transported via underwater pollinators. So similar to how pollen sticks to things on land, we're seeing the same sticky pollen attaching itself to these underwater creatures. And the first study that reported this was written by Van Tussenbroek et al. In 2016, where they did some experiments in Mexico to show this pollination in action. They also demonstrated that pollination is likely more common at night, because a lot of the organisms that tend to be pollinators emerge from the sediments at night. They come out to feed and reproduce and do other things in the evenings. And those are the things that tend to be pollinating. And a lot of the flower species actually send up their flowers at night too. The flowers open at night. So next I'm going to talk about the 6 different species of seagrasses that we have in Trinidad and Tobago, and go into a little bit on their morphology, what they look like, as well as their flowers and the different kind of phases of their flowers. So we have turtle grass, which is Thalassia testudinum, shoalgrass, Halodule rhytii, paddlegrass, Halophila decipiens, and these top 3 species are the main ones that we found flowers for this study. So they'll have a little bit more detailed information. And then we have clovergrass, Halophila bailinus, manatee grass, Syringodium filiforme, that's only found in Tobago, and widgeon grass, Rupia maritima, which is found mostly in more brackish or freshwater, right? So for these last 3 species, I've mostly just kind of pulled stuff on their flowers from the literature. Right, so this first species is turtle grass. This is the main dominant climax species found in Trinidad and Tobago. It's very thick-bladed, it grows quite tall, and it's very deep roots and rhizomes, very thick rhizomes. It's really the best at kind of doing all those ecosystem services that I mentioned earlier. It's really good at doing all those things, right? So we have in the picture here some of the flowers. So on the right here, we've got male flowers here and the female flowers next to it. So the female flowers are a little stringier, the male flowers are a little thicker petaled. And then when the male flowers come out, they have, like I mentioned, this mucilage. So you can see in the upper right here, this is one of the flowers we collected at night. And when I pulled it out of the water, you could see the mucilage just in like kind of a big bubble. And if you look and see the little white dots in there, those are actually the individual pollen grains. So you can see them with the naked eye. And the pollen looks like this here. So from what we've seen under the microscope so far, it looks like the pollen may kind of hydrates before it's released. So a lot of the pollen just looks like the dark kind of round spheres here. And then we think once it's ready to open, that it adds either a layer of water or maybe a layer of like fats or something is being added to the outside because it's not present on all the pollen grains. And that may facilitate it floating in the water, or it may be sticky. It may facilitate the pollen actually sticking to pollinators or sticking to the female flower. Not really sure. Here we can see once the female flower is pollinated by the male pollen, then it develops it immature fruits. Then the fruit matures. It looks kind of like a spiky little ball. And then once that opens, you have two little seeds inside, which you can see here. I mean, kind of, I guess, kind of big seeds, not super little. So these flowers are about like 2 centimeters in width. They're fairly small, but the biggest of our seagrass flowers. So they're the easiest to find. Okay, so this is just a picture to show the differences in flower morphology that we've noticed between Trinidad and Tobago. So the flowers on the left here are ones that we saw in Tobago. The one on the left is one that just came out of the bud. And so it's looking all sticky and full of pollen. And the one on the right here is a little bit older. Its petals are probably going to start falling off soon. It's kind of on the later stages of flowering. Right, so that's the typical colors and kind of whitish we see in Tobago. But the flowers, the male flowers in Trinidad are very like pink and magenta color. They're really, really pretty. And from the research I've done so far, don't really see this pink color in most of the rest of the Caribbean. They typically are more this white color. So we're really kind of interested in seeing what's going on here and what's leading to that difference in color. All right, so moving on to our next species is manatee grass. Manatee grass is another late succession species, so it exists in very stable environments. It's only found in Tobago, and it kind of looks like spaghetti strands. It's a cylinder-shaped grass, and this helps it deal with higher currents, so it has less drag on it. So it can kind of grow in areas with a little more wave action than the other species. Yeah, so they're flowers that are grouped in inflorescence, and this inflorescence is called a cyme. So you can see the male cyme here in the upper left, right? So they're formed on separate plants. This is a dioecious species as well, and each cyme bears flowers at different stages of development. The female flowers and male flowers you can see at the bottom here. So this is an open male flower. This pollen release picture would be just as it's kind of coming out of the cyme is when a lot of pollen comes out. And then it would be fully open in this picture here. And this is the female flower. And then once the female flower is pollinated, then it turns into these fruits. So you can see here, and then the fruits have seeds. And once the seeds start growing in the ground, then they look like the seedling here. And this is just a picture showing the pollen and where it's found on the male flower. All right, so our next species is shoalgrass. So this is a very early successional species. It's a colonizer. They're very tolerant to kind of poor conditions and shallow areas that get exposed to air. They can deal with a wide range of temperatures and salinities. It's kind of like the weeds of the seagrass. So they have very thin, flat blades and kind of puny rhizomes and roots. They don't go particularly deep. But they're great for getting an area ready for those colonizing species like the turtle grass and manatee grass to move on in. They have very discrete flowers that are quite difficult to find. So the male flower, when it first emerges, is kind of white in color. You can kind of see it at like the fattened base of the leaf. And then it comes out, it's white. Or kind of clearish, often with a bubble, which we're not really sure what that's about either. And the pollen comes off, and after most of the pollen's gone, it starts to look more kind of brownish in color, like you can see here, and then just gets kind of darker and darker as the flower's out, which looks really, really similar to the snails that like to chew on it and feed on them. Right, here we have a male flower, looking at it under the microscope where you can see kind of all these filaments coming off is the pollen. So it's in these like long stringy fragments. So that's zoomed in even more. Here we have the female flowers, which are even harder to see. This is zoomed in quite a bit. Basically, they're just two little threads, two little pistils that come out. Very, very thin, they're white, really hard to see, but we found a couple of them, but I'm sure there's a lot more out there that we just didn't find. And then here you can see the young fruits, and then a larger, but still young fruit. And then these next two pictures show a mature fruit, which has kind of like a hard, exterior shell on it, and then the seeds inside. So again, this species is also dioecious. Our next species is the paddlegrass. This is a very small species. Again, early successional. It's quite tolerant to poor conditions. This species is kind of different because it's actually monoecious. So the male and the female flower are on the same plant, and they actually grow at its same base of leaves, right? So here's the little paddle-shaped leaves. They grow in pairs. They're quite tiny, not any more than like 3 centimeters in height. And the flowers, as you can imagine, if the leaves are that small, the flowers are just teeny tiny. You can kind of get some perspective in this picture with the male flower here of how tiny these things really are. So again, really hard to see, but we've managed to find quite a few of these just because they're pretty common. Like often you have to pull up the grass a little bit to actually see them, but they are there. So the flower buds, they're kind of both the male and female flower are in like the same kind of capsule, right? And the buds then come out of that little capsule. And here you can see it kind of opening up, and then out come the two flowers, male on the left, female on the right. And then when the male opens, you get a immediate like pollen release. And then the flower emerges, which still would have pollen on it. And the female flower you can see next to it. And then just on the bottom here, I have some more pictures of males and females. So the male here, another one here with, you can kind of see a little bit of female in the front too. And then we've got the female flower here and here. Oh, and the pollen for this is very tiny, but it's also spherical or circular in shape, as you can see in this image here, but very, very tiny. And this is what the progression of the fruits and the seeds look like. So this is a young fruit, an older fruit. This is a fruit that has seeds in it. And this is what the fruits look like at the base of the plants, although that's quite zoomed in. They're pretty tiny. All right, our next two species, I'm gonna go over fairly quickly. We have the clovergrass, and this is a new species that we found in Trinidad and Shikari a couple of years ago. And during these surveys, we actually found it in Bonacord Lagoon in Tobago as well. So these are dioecious again. So either a female or a male flower comes out at the base of this clovergrass. It's called clovergrass. It kind of resembles four-leaf clover. It's very similar to the paddlegrass, except instead of 2 leaves coming up, it has 4. That's really the main way you can tell it apart. It's a little smaller and a little narrower leaves than the paddlegrass, I would say. But otherwise, it looks almost identical unless you count the leaves down to the base. So the flower comes out the middle here. So you can see a male flower here, an open one, and a female flower here. This is what the flower bud looks like, and then it would release all this pollen. And then you can see it has 3 anthers associated with each of its 3 petals. And the female flower, it looks very similar to the shoalgrass, and it just has these kind of 3 thread-like stamens coming out. And then once the female flower is pollinated, then it would produce a young fruit. And I don't have any pictures of what the more mature fruit looks like, but just a bigger version of that, I'm sure. This is also an early successional species, more tolerant to poor conditions, similar again to the paddlegrass. And the last species we have is widgeon grass. This is one of the hermaphroditic seagrass species. It's highly reproductive. So each inflorescence would have 2 hermaphrodite flowers, and the flowers are composed of 2 pairs of anthers in the horizontal position and 3 to 4 pistils. Right, so here you have an immature flower, a mature flower. Here you can see the anthers opening, which are these paired things here. So it's 1 flower on the top. Oh, sorry, I'm pointing to the wrong thing. Immature, mature, anthers opening, and here you can see the 4 anthers of 2 flowers. So 4 anthers on top, 4 anthers on the bottom. So there's 2 separate flowers, and then these little green things poking out are the pistils. And you can see it better here when everything's all open, little green pistils coming out. And each basically flower would produce a single fruit once it's pollinated. This species looks really similar to shoalgrass, except it kind of branches out. It's flat, it's thin, but you don't see this kind of bract system in shoalgrass at all. It just goes straight from the ground, whereas this will kind of branch off as it grows. which is kind of how you can tell it apart. And like I mentioned before, this mostly grows in brackish to freshwater. And one more species I just wanted to quickly mention is this invasive seagrass species that's been found throughout the Caribbean, Halophilus stipulaceae. It's basically everywhere around us, but we haven't found it in Trinidad and Tobago yet. It was introduced in 2008 from the Red Sea and the Indian Ocean. It's also invaded the Mediterranean first and then came across to the Caribbean. I think via the yachting industry. So these grow in pairs, flat-bladed, thick, kind of similar in thickness to the turtle grass, but they're not very big. I would say about 5 centimeters long. So they're bigger than the paddle grass, but they don't grow any taller than that. Okay, so for this project, we wanted to do a couple things. First, to determine when flowering occurs for this different seagrass species in Trinidad and Tobago, and to try to identify what may be triggering flowering to happen. And then we also wanted to observe, capture, and identify potential pollinators using video cameras, light traps, flower samples, and microscopes to view what we find. So for our methods, we surveyed seagrasses and pollinators mostly after dark in Williams Bay, Trinidad, and Bonacord Lagoon, Tobago. And we did this on a weekly basis since July of last year. We also deployed some semi-permanent loggers at both sites to record light levels and temperature, which is shown up here. This is our HOBO logger, and down here below it is our salinity logger made by Star Audi. Then we used vials, as you can see here, to collect flowers and their visitors. So the vial was placed over the entire flower, and then the flower kind of pulled up and the cap put on the vial to try and keep everything contained in there when that was possible. We also used cameras to film the flowers and their visitors, and these filmed for between like 15 and 20 minutes. We set light traps over the flowers to catch potential pollinators. So the light trap basically works like it's a funnel underneath, and the light is at the top shining down. So that light attracts the plankton into the funnel. And then the thought is once they get into the funnel and into the rest of the trap, then they can't get out very easily. And so we would then, once we collected those, then we would filter them, preserve them, and before looking at them on the microscope, we would add aniline blue dye to highlight the pollen, because that tends to dye really well with the aniline blue. And then we would identify the organisms that we found and check each of them for any potential pollen and take pictures of them. Okay, so what did we find? We found that turtle grass flowering may be triggered by the start of the rainy season and warmer waters that occur from April to July. In Mexico, they also found flowering that started in April but extended into September. So I'm just going to skip to my graph so you can see the turtle grass here is the flat lines. So this is flower presence per diver visits. Trinidad on the top and Tobago on the bottom. If you look at the horizontal lines, shows you the turtle grass. And for the shoal grass flowers, we only saw those in Trinidad. We saw them in 2022 in June, July, and September. And then in 2023 from February to May. So very different time frames. So we haven't been able to identify any kind of triggers for those flowers as yet. In Mexico, they found the flowering was from March to August, which is kind of similar, but not, the overlap's still not exactly right with what we were seeing for this year anyways. So yeah, a lot to figure out with that species. And you can see the shoalgrass is just in Trinidad. So it's the gray bars are the shoalgrass, the Halogili. So you can see kind of how variable their flowering was. And for whatever reason, we didn't find them in Tobago, the flowers in Tobago at all, which was odd. All right, so if we look at the paddlegrass flowers, they were seen from late February to early May and in July in Tobago, and similarly from March to mid-May in Trinidad. But also we saw them from July to September of 2023 in both places. So they seem to be doing a lot of flowering in both Trinidad and Tobago. And in Mexico, they saw the flowers from May to October, which kind of overlaps somewhat with ours, but not completely. So we can see that here. That's the diagonal lines are the paddlegrass. So we can see mostly in Trinidad, March to May, and then June to September. But we didn't see them in 2022 during that time, which was kind of strange. But it could be because we didn't have like a super rainy season this year. So maybe that's why the flowering was able to continue. And we actually didn't see the paddlegrass. I forget which month it was that we started seeing it, but we couldn't find any paddlegrass in Trinidad for several months. And then it kind of appeared. And once it appeared, it started flowering. Whereas in Tobago, we saw the flowering kind of in July, and then flowering again, February, March, April, May. Nothing in June, but then July, August, September. Paddlegrass does seem to be somewhat seasonal in Trinidad, and it may not be able to tolerate low salinities. The clovergrass we found in Tobago waters for the first time during our surveys, which I mentioned earlier. And their flowers potentially occurred in early March and early June. There's a couple of records of that, but don't have any pictures to confirm it from my Tobago crew. And this kind of coincides with when they've seen the flowers in Belize on the same kind of general time frame from April to May. We didn't see any flowers in the manatee grass, which was very odd, and no flowers of the shoal grass in Tobago. For the manatee grass in Mexico, they found their flowers to occur from March to July. So we might try and do some more extensive surveys during that time in Tobago this coming year to see if we can find some flowers. It's also thought that they flower during the day, so that could be part of the problem why we didn't see any flowers either. But you would think we would see some fruits or something, because the flowers might not last long, but it takes a little while for the fruits and stuff to develop. And then the clovergrass and manatee grass weren't present in Williams Bay, so we don't have any data on those. All right, finally on to the pollinators. We found several species of small snails, amphipods, shrimp, polychaete worms, and small fishes that were seen visiting turtlegrass and shoalgrass flowers, as well as, I feel like I'm missing some other things in there, some hermit crabs, essentially some larger snails. We saw a lot of stuff on these flowers, which is really cool. We saw that some of the copepods, nematodes, and tineids, excuse me, from our light trap and flower samples had pollen on them or in them. and either in whole or fragmented form. At least it looks like pollen when we observed it under the microscope. And I'll show you guys some pictures of this in a second. So I'm going to show this, these pictures. These are clips from some of our video data. So on the left, you can see a shrimp, a small mysid shrimp maybe, visiting the male flower. And on the right side, this is a female turtle grass flower, male turtle grass flower, female turtle grass flower. And we see what looks to be the same species on the female flower as well. So this species seems to visit both flowers, which is a pretty good indicator of pollinating, right? So it visits the male, gets pollen, and then comes and visits the female to pollinate it. These are a couple species that were quite predominant around the flowers. This small little snail species, as you can see on the shoal grass male flower here, as well as we saw tons of these tube amphipods, which are super cool. I didn't even know they were a thing before doing this research. But yeah, it's a little crustacean that lives in this tube, and they move around. They can go in and out of their tubes, can kind of swim even in their tubes. It's quite fascinating. But yeah, they really like to visit the flowers, both the shoal grass and turtle grass flowers. And these are a couple pictures of some of the species that we saw with potential pollen on them. So on the left, we've got Paracalenus species. We weren't able to identify it any further than that at this point. But you can see this little blue dot inside, so it may have eaten some pollen. And then we can see on the right, this copepod nauplii with potential pollen on the right side here, as well as some potential pollen in its antennae, which is cool. If we look at the former study done by Van Tissenbroek et al. In 2016, they found very similar species to be pollinators of turtle grass flowers in Mexico, lots of crustaceans and polychaete worms. It didn't really get into the polychaete worms from our site, but they were extremely abundant, especially on some nights. We didn't catch too many of them in the light traps. They didn't really seem to go in the light traps for whatever reason. So we weren't able to get much information on whether they have pollen on them or not, but they definitely are throughout the water column and visiting the flowers in our videos. As far as the crustaceans that Van Tissenbroek would have seen, visiting flowers were things like isopods, copepods, ostracods, mysid shrimp, tanaids, humaceans, and zoea, which are larval crabs. And they also documented 3 species of polychaetes across 3 different families on the flowers. And below this are some pictures of some of these individuals with pollen on them. So you can see the zoea larvae in these first 2 images with the turtle grass pollen inside and outside. And this kind of on the abdomen or the tail, I should say. And then this is one of the polychaete worms with the pollen, a piece of pollen stuck to its parapodia. Seagrass genetic diversity could be reliant on these pollinators, and thus we really need to consider them when we're managing and restoring seagrasses. And in the same stance, the pollinators may be reliant on nutrition from the flowers, particularly the mucilage, maybe even the flowers themselves. So to have higher diversity of pollinators, we need to have flowering seagrasses and vice versa. And really, we just need a lot more knowledge on seagrass flowering, what's causing these seagrasses to flower, and the pollinators that are most associated with each seagrass species. And once we have this data, then we'll be able to better recover and sustain these important ecosystems. So we saw the turtlegrass male flowers open at night, which is also when these crustaceans and polychaete worms emerge to feed from the sediments. And the flowers bloom near the base of the seagrass leaves, close to the substrate, which gives these critters kind of easy access to the sticky pollen, especially for things like snails and stuff that are crawling along. And it's really likely that there's many other types of pollinators for seagrasses, including species that are active during the day, because a lot of these flowers persist, like they bloom at night, but then they persist even for several days. So pollination could still happen during the day following that initial bloom. And some of these pollinators could even be megafauna, like green sea turtles. And because we have seen in other studies, Tola et al. 2017, that these megafauna can transport seagrass seeds. So if they can transport seagrass seeds, it's highly likely they also could transport pollen. And with that, I am happy to take any questions. Thank you so much for listening. If you do happen to see any seagrass flowers, they're kind of hard to see other than for the turtlegrass species, but now that you know what to look for, these teeny tiny little flowers on the other species, let me know where you see them, the date, the time of day, what species, and the depth, and we can add that to our database. It would be lovely. And also be on the lookout for this invasive seagrass species seen here, so we can try to keep it under wraps. When it gets here, I'm sure it's just a matter of time. All right, thanks.
Thank you very much, Dr. Kingon, for that very interesting presentation. I think the presentation's always brought to life when we have a lot of photos, so we thank you for that as well. We are now happy to open up for the Q&A. I'm not seeing any questions there as yet. If you have questions, you need to type it into the Q&A. I have one question while we're waiting for attendees to put their own. I'm very interested in that color difference that we're seeing in the turtlegrass flowers, is it? And I was wondering, Dr. Kingon, if perhaps there are environmental conditions at that particular site in Trinidad that may be causing it, or whether there's a subspecies. What do you think?
Yeah, I mean, we've been talking about trying to do some genetic analysis across these two regions as a first step. But the water conditions are very different, as I think all of us know, between Trinidad and Tobago. So it could be just an environmental factor that's leading to it, or it could be an adaptation to, look like something that's, amongst the seagrass in Trinidad. We really don't know at this point, but it is quite interesting. But yeah, that brackish water that we get in Trinidad, I think must play some kind of part, either on what kind of organisms are present that might have led it to be that color, or something that it just incorporates into the flower because of those environmental conditions. Not really sure.
Well,
yeah.
Definitely with the further investigation.
Sure.
We have a question or a statement from Akila. Akila is saying, nice research. Lots of hours involved in this for sure. Did you do any nutrient water quality testing as well? And if so, any findings from this or any info from the literature on the onset of flowering?
So we didn't analyze nutrients for this study. There's some data potentially for in the past. But yeah, that would definitely be something to look into, because it very well could be a nutrient influx that leads to some of these flowering patterns. Yeah, so that would be something to look into further for sure.
We have a series of questions from Justin. Okay, so Justin, the first question is, what is the feasibility rate? the feasibility rate of the seagrass growing in other areas around TNT? I'm not quite sure about feasibility rate, but that's question one. Question two, does tourism, fishing, or recreational use affect the growth of seagrass? And thirdly, how extensive is the soil and coastal preservation regarding seagrass? So Kelly, maybe you would have to look at the Q&A yourself so that you could recap the three questions asked.
I got it pulled up. So the feasibility, I guess he's asking of the seagrass growing elsewhere in Trinidad and Tobago. So seagrasses were much more plentiful in both places. And a lot of the issues with seagrasses here and them disappearing has to do with coastal developments. So when you have coastal developments, there's a lot of sedimentation. And essentially those sediments just kind of bury the seagrasses. And if those disturbances continue, then the seagrasses aren't able to recover on their own. So once you've fixed some of those issues from humans and our impacts on the coastal area, then really the seagrasses should come back on their own. And we actually saw that in Williams Bay. All the growth that's there now is new growth, because with the development of the water park and all the stuff that's going on by the boardwalk there, the sedimentation from that actually covered the entire seagrass bed. And I think it was 2014, around then. And so we've kind of been tracking the recovery of the seagrasses, because once construction stopped, then the seagrasses were able to come back on their own. We haven't done any kind of restoration at all. It's just because the issue was stopped, and so the seagrasses came back. But I think in other areas that issue continues as we develop the coastline. So it's really more about controlling human actions and trying to think about how to fix those issues. As far as the second question, does tourism, fishing, or recreational use affect the growth of seagrass? Yes and no. It depends how it's done. And there's really, I don't think that many good studies like documenting those impacts. But I would say just from experience, like in Williams Bay, where people bathe a lot and are walking continuously on the seagrasses, that the seagrasses aren't very present in those areas. So you have to kind of get away from the main bathing zones to find seagrasses. So that doesn't help. Also, some people will actually pull it up because they don't like it on their feet. Or in some cases, they just pull it up because they don't want it to be there, like at Nyla Pool. That's a whole nother issue. So that can be a problem, but I think if people start to appreciate seagrasses more and all the benefits that it provides to us, that hopefully we'll be excited pull over seagrass and things like that, because it's really exciting and fun, rather than try to get rid of it. As far as the third question, how extensive is the soil and coastal preservation regarding seagrass? I don't know if I know exactly what you're meaning, but really conservation for seagrass is extremely lacking, especially in Trinidad and Tobago, but across most areas where it grows. Yeah, and it's really not protected at all as far as I know, other than in Bonacore Lagoon, which is a protected area in Tobago. It's not protected. And the rest of the Buku Reef Park there. I realized I forgot about mentioning fishing and its effects on seagrass. That could be a possibility depending on what kind of fishing gear is used. But if it's just hook and line, probably not going to have that big of an impact on seagrass itself. But there's always ecosystem considerations as well. If you remove too many of the fishes, then it's an important component of a seagrass ecosystem.
Okay, thanks very much, Dr. Kingon. And I'm happy that you gave that information about really what's happening at Williams Bay, that perhaps closer to the shore, there may be less, but where you sampled, which is a bit further out, that you were able to get seagrasses, because we're quite aware that the Institute of Marine Affairs is also doing work on seagrasses on their marine project in Tobago. There is some audiovisual material that we've been seeing on television that speaks to Williams Bay. And so I think it was important for you to explain the differentiation of what's happening close to the shore and what's happening a little further out, so that people don't go away thinking that we were fabricating that there were seagrasses present at Williams Bay. Daniela has a question. I just want to get through the two questions that are remaining, given our time. Daniela is asking, are there any other areas within Trinidad that you would want to repeat this study?
Yeah, I mean, wherever there are seagrasses still around Trinidad. Originally, we had wanted to do some surveys around Shaka Shikari, because that's where we found the clover grass, to try and look at their flowering. But logistically, to get there at night, it was just kind of hard to organize that. But I would definitely say down the islands and trying to visit some of these other areas that historically had seagrasses, to see what's going on there, like Claxton Bay and off Guayari. Those are other areas that definitely need some more investigation to see how the seagrasses are doing there and if there's anything flowering as well.
Okay. And Justin is asking, would replanting programs work better in rural areas? I guess he means areas where there's less coastal development. And are you open to working with local NGOs interested in seagrass, I guess, replantation programs?
So, like I said before, I think the main issue with, or are you guys still hearing me? Internet said it's giving trouble. Okay, good. The main issue with restoring seagrasses is human impacts. And if you don't fix the human impacts, the seagrasses aren't gonna come back. Even if you replant them, they're just gonna die again. So I think really focusing on fixing those issues is much more important than trying to replant seagrasses, where naturally they should come back on their own. I think if you realize, you fixed all the human influences in the area and the seagrasses still aren't coming back, you could try restoration of replanting some seagrasses, but it's always best to start with those colonizers, just like nature intends, and use shoal grass and paddle grass and things like that, which are a little harder to move and deal with, but you can also potentially collect seeds and things like that. That those would be the best option for restoration. And yeah, definitely happy to help with any kind of restoration and seagrass work, for sure.
Okay, I did say two more, but I'll take the last one. Akila is asking, so would your research therefore try to work towards protecting the seagrasses directly, given its small areas? I guess that we have so few.
Been in discussion with people in Tobago about really trying to conserve their seagrasses especially in the Buku Reef Marine Park. That's supposed to be, protected from our practices, but they're really key, not only for their own functions, but they also take a lot of the nutrients out of the water that are really bad for the coral reefs that are just outside of where the seagrasses are. So if you remove those seagrasses from places like Nylon Pool, then you're putting the coral reefs further out at greater risk. And so you don't want to do that. So the seagrasses are good. We should be happy that they're there. The nutrients pollution going into Bonaparte Lagoon should be better controlled. That's an issue that needs attention. As far as in Trinidad, it's a little trickier. I've spoken to CDA about the options of this, but they claim they don't have any jurisdiction over the water itself. So it's kind of like who would implement that? Although they have been doing some activities on the water. I've noticed they've stopped fishing, like along the boardwalk in certain areas. So I think they have a little more jurisdiction than they lead on to. So, but yeah, more stakeholders we can have involved and kind of go to them and other ministries to see what can be done would definitely be a good thing.
Great. Okay, so with that final response, we want to wrap the session up. On behalf of the BES-Net TT team, yes?
Sorry, if we have just another minute, I had a couple of little short clips of the pollinators on the flowers, if you don't mind if I just show them.
Okay, sure. We can go ahead with them. And audience, feel free to stick around for this.
Yeah, if you don't watch, sorry.
Go ahead.
I couldn't get them into my presentation, so let's see if we can get this. So in this first one, there's a worm that you can see wrapping around the flower there. And then here we have a fish, a little mohara or blinch that goes kind of right up to the flower. It kind of brushes against it with its head first. And then it kind of goes around and then it comes back in and kind of moves its tail, its back half of its body around the flower. You can also see there's a little snail down here that may or may not crawl up onto the flower. So that's a turtle grass flower. And then the other one I have is of a shoal grass flower. Play. Wrong screen. Sorry about that. Put it on the right screen here. All right, so this is the male shoalgrass flower here, and you can see these little isopods and a polycute worm rubbing against it.
I think you need to share another screen because we're not seeing that one.
Not seeing this one. What does it say? Oh, it says screen sharing is paused. Maybe I need to share again. Okay. Straighten up thoughts. Seeing it now?
Yes, we are.
Okay, great. All right. What else here? Just saw the first one. Okay, we got little amphipods here that's crawling on the water, on the flower, and then a polycute worm comes and like rubs all up on it as well. Other little worms in the picture. And then like two snails that come in. They don't actually climb on the, actually 3 snails. They come in. They don't climb on the flower in this video, but on some of the others, you can actually see the snails on there. And that last kind of big worm, red worm that went by it. So, yeah. That's all I have. Thanks, Celeste.
Okay, great. No, I think our audience would have appreciated the video. The still photos were great as well. So thanks very much, Dr. Kingon, and please extend our thanks to your team also. Members of the audience, I thank you for your interest and your participation in today's webinar. Please remember that the recording, as well as any responses to any unanswered questions, will be posted to the project Facebook page, and the whole webinar recording will also be carried on our YouTube channel. So we look forward to your continued interest and engagement in the BES-Net TT project. Keep following us on Facebook so that you can learn about any new webinars that we will be sharing with you. Have a good afternoon and have a great day. Goodbye. Thanks, Kelly.
Thanks, Les. Thanks, BES-Net. Thanks for coming, guys.